Let's see if I can. Yeah, that should be okay. Quiet, so
Hi everyone, my name is Peggy. I'm from UCL and ZSL UK, University College London. And my project was trying to ask if we can infer ecological health from unlabeled data using an unsupervised learning method. So why am I asking this question? There we go, just building the suspense. So we're losing biodiversity at alarming rates. Vertebrate populations, for example, have gone down 70% since 1970. And this is despite numerous global interventions, like the Aichi targets, the Convention of Biological Diversity. All these things are trying to reduce biodiversity loss, but it's still happening. And it's really clear that we need a really strong understanding of how environments are responding to human disturbance and how conservation interventions are impacting those responses. But just because of the speed that we're losing biodiversity at, we need to do this as fast as possible. And well, the reason why most of you are here is that a lot of conservationists are moving towards using conservation tech to try and do this and speed up this process. So my PhD is focused on a field site with a huge data set from a field site in Nepal. I'm part of the Biome Health Project, which is a wider project with field sites in Kenya, Fiji, Borneo, and I'm doing the Nepal site. And all of our field sites are specifically designed so that they cover a gradient of increasing human pressure. And each biome has a different pressure, and the one that I'm specifically looking at is fragmentation and urbanization. So the field site, each little dot here on the map is a camera trap site. So they're in like 2 kilometers radius. And the gradient goes from sort of north to southeast, where we've got the national park within the red boundary, the buffer zone, which is semi-protected land, and outside the buffer zone. So as you go from north to south, you get like increasing levels of fragmentation, agriculture, and livestock. And it's a really exciting field site because we've got some amazing animals. It's really high diversity. We've got rhinos, tigers, elephants, leopards, loads of different types of antelope. And we're using camera traps and acoustics. But in this project, I'm specifically looking at the camera trap images. So for the most part, we've been manually tagging our camera trap images, and this is really, really slow. So at the moment, we've labeled about 50,000 images, and that took probably more than 3 months with 3 people. And as you can see from this middle image, we make many errors. So this is like an attempt at us making a classifier, and the machine thinks it's a cheetah, which is a deer, and it's actually labeled as a goat, but actually it's a human. So so much confusion there. And eventually, what I'm interested in my research is to understand overall community and of wildlife and how that changes. I'm not looking at any individual species. So I was wondering, is there any way that we can skip the labeling step and go straight to the metric? And this was inspired actually from the acoustic world, where there's been some work on this to look at the feature embeddings of a model that is not trained on your own data set. And this is some work by Sarab Sefi, and it's showing that these are all like, each dot here is an acoustic file, and if you color it, and sorry, the model like spits out these embeddings and you can map it out, and if you color it, it kind of groups by above ground biomass. So that's suggesting that there's some kind of information within this sound file without the machine even having to know what is in there already. So I was wondering if we could do this for images as well. So my general approach was to put images through the mega detector so you can get the bounding boxes of animals. For my work, I didn't do this because we manually labeled it. Determine the features, and like what I mean by that is when you put your images through a classifier, it will go through a feature extractor, and it will like turn your images into like really long vectors. And then normally you would then put it through a classifier, which will work out how those features are correlated with each other, and therefore, is it a tiger or is it a deer? But I'm just cutting off that classifier bit at the end and looking at the features. And it's actually 2,048 dimensions, those features, but it's a clever thing called UMAP to shrink that down into 2 dimensions so you can put it on an X and Y axis to visualize it. And then my hypothesis is that if you have a site with more different species in it, the distribution of those features will be more clustered. So maybe like the clustering will be correlated with species richness or any other kind of environmental metric. So that's what I was testing. So I split my data into like a train test eval site, and I did that by geographic region because I wanted to apply this method to a new area. So I made sure that my like test site was like a completely different area that my train set that my method hadn't seen before. So how did it work? This was like a first pass, like passing all of my images through the model. And so this is like how they come out distributed on the other side. And this is just like a standard ResNet 50 trained on ImageNet. ImageNet is like a huge data set of like loads of different images and objects. And so like on the left, it's colored by time of day, and on the right, it's colored by management zone. So like it's the darkest is the buffer zone, which is like the middle area, and then national park is like the most highly protected. So you can see that mainly it's like split by time of day at the moment, but there are a few little groups like coming out. And I was also interested in what those like ones around the edge are, like these random ones, and I thought it'd be something really interesting, but it's just a cow. Some like more digging that needs to happen there, like why this cow is so out of the I'm not sure yet. So yeah, my question was like, do the distribution of these embeddings link to any kind of environmental metric? And to start off with, I looked at species richness, because from our already labeled camera trap data, we know that species richness is highest in the national park and significantly lower outside the buffer zone. So it does kind of move in that gradient. And this histogram is showing that like this is like the actual distribution of how many species we see at each camera trap site. So it's kind of normally distributed with like on average 7 species. So I ran my, I did like a k-means clustering analysis on this and tried to find the optimal number of clusters using this metric called a silhouette score, which I just learned about this week. And yeah, so technically I should get like the optimal number of clusters per site. And if it works well, it will be like the same distribution as this. So does it work? No. Like this is the number of clusters, and it came up with like almost consistently 2 clusters with like a few extras going out. But I mean, I was never expecting to get the exact number of species as is the number of clusters. So what we're actually interested in is the order. So is the site with the highest species richness also the site with the highest number of clusters? So we could do that by looking at the residual error. Also, no, it doesn't work because so the residual error is showing like if the error is 0, then it's like guess the species richness exactly right. But mostly it's under predicting, and that's because of this high number of 2s. But you know, if it was only under predicting or only over predicting, then you could say it's kind of always going in the right direction. But sometimes it's like going both ways, so it's not very reliable, whatever is happening there. So how can I improve this? Firstly, I tried to have a look at the clustering method, and I tried changing the number of dimensions. So as I said, my feature was started with 20,048, so I'd like tried to reduce that to see if that made any difference, and it didn't really seem to. These histograms are that residual error plot again, and it's like not really changing the distribution that much, although the x-axes are different here. I didn't have time to fix them. And I also, like, with help from Eli, made this little rank score thing. So like technically if the highest site for species richness is also the highest site for clusters, like, and it goes in the same order, that value should be 1, because that's like perfect score. and 0 is rubbish score. So I got 0.3, so it's not doing very well, basically. But like this seemed to actually get worse with the reducing the number of dimensions. So yeah, reducing dimensions doesn't really improve anything by the look of it. And then the next thing I need to do is look at nearest neighbor distance, which is another parameter which I still have to do. So the second thing I wanted to test was, can I use a new model, a different model? So I used a model that was borrowed from a colleague who created a self-supervised model on another data set from the Bio and Health Project, which was in Kenya. So I thought maybe because it's camera trap data, it's similar, it might pick out some more things, but it actually made the embeddings are kind of like more grouped into one thing, and there's less clusters. You can still see that like day-night grouping there, so but like not really much by species. And this is just a picture of a camera trap in Kenya. This is the things that we experience. And this is the same thing again, of like the dimensions don't really change. And
The ranks
these are wrong. These are wrong. This is the right number for the first one. Yeah, I forgot to change these, but yeah, ignore that. Yeah, but so the rank score is slightly higher for this one, so but like marginally. And then my next step is to try and reduce that variation in training data. So there was like always 2 clusters, almost always, and like it's pretty obvious why that is. There's quite an obvious split in camera trap data between day and night. So I tried to remove the nighttime images to see if that did anything. And so this bottom cluster is like the daytime, it's mainly nighttime images. And it brought out a few species as like separate clusters. So I think that there's like something to follow up there. Just to explain this, the colors are different in these graphs. So the species don't match, which is really annoying, but yeah, just look at the clusters. Yeah, so and then there's a few more things to do, like trying some different clustering methods. K-means isn't that great because it's like considers clusters to be in a kind of a spherical shape. So there's these other methods like HDV scan, which look at density rather than shape. So I'm going to try them and see how that comes out. Yeah, and then how am I going to use this in the future? So my lab and people that I'm surrounded with in my field are all about using tech for biodiversity monitoring, and there's loads of exciting things happening. Like this is a microphone connected to a Raspberry Pi that I made with a friend, and we downloaded the BirdNet algorithm onto it, and it's now sat in my garden telling me, and it's like connected to the Wi-Fi, and it's telling me what birds are in the garden. It's like incredible. And then we've got my supervisor Kate Jones has her bat monitoring project in London, and the colleague I mentioned earlier, Sarah, is doing the same thing in Norway, of like doing this amazing acoustics monitoring. So that's like the aim, is like if we can move towards these awesome like monitoring methods that are really automated, you can put them places and you don't have to teach people how to code and spend hours tagging images and things like that. So I really hope that this kind of thing moves towards that kind of metric for camera trap data. And yeah, it's quite exciting to me to bring all this information together. So yeah, thanks everyone.
That's a monster.
What's happening? Awesome. Questions for Peggy?
Yeah.
So the clusters after you took out the nighttime, those like more kind of defined ones, were they like, did they tend to be just like the most common animals? Or
So I think to be honest, like a lot of them are, it's like a problem with my data is that I'm like overwhelmed with livestock images. So there's so many cows, so many sheep. So like, for example, like this purple area and this sort of beige area, that's buffalo, I think. So like they're in the same area, and like the overwhelming mass here is just cows. But I think that this one that came out is Might be sheep, actually. So yeah, you're right. It's like not that, but yeah, I think it would be really cool to find a way of like dealing with those like non-wild animals and seeing. I mean, I think there's so many more things that I'm going to try with this and like poke, and so I'll take out the domestic animals and see how different it looks. But yeah.
Yeah, Alex.
Do you have any idea why the cow butt may have broken or like moved so far away out of your cluster? I'm really intrigued by this image.
So I have a confession to make, which is like the actual picture that this was actually a cow butt, just the bum. and then like I did a load of coding to find out which one it was, and then it was like the next day and I couldn't find it again, so I did this nice picture of a cow. But yeah, the actual cow was like the same color as the background, so I really have no idea, because like there are so many cow pictures and it wasn't that out of the ordinary. But yeah, I think it was probably the color of it and the color of the background. But yeah, I'm definitely going to look more into it. See why. Yeah.
Do you think if you took like something like eBird data or iNaturalist, and you got just like photos of each bird species or each, whatever animal plant species from iNaturalist, and you did the same sort of thing, would you expect to get the same sort of result, or would you expect a different result? Or
Do you mean using my data or using the
just using
the
like
iNat data on this model?
yeah, exactly. Yeah, grabbing iNat photos or even eBird stuff, just grabbing photo. We all just like go to Google and get it and throw it into the model.
See what it looks like. It's a good question. I don't actually know. I imagine the eBird data is like kind of cleaner and nicer, so it might like all the images of the same bird might be similar than my data, which is like every animal is in very different like position and looks different. So it might show more interesting clusters, but it would be a really interesting thing to look at. And one of the other things I wanted to do was use the iNaturalist model that they use on my data to see how that kind of predicts where things go. Does that answer your question?
Yeah, cool. Thank you.
Yeah, Ethan.
So these like embeddings that you get are super interesting from like standpoint of like, yeah, you take an image and then you like turn it into this continuous metric. So I'm wondering if you've thought about like maybe some kind of like weak supervision with remote sensing data and trying to bring like these multimodal I mean, yeah, I don't know. I could just imagine a scenario where like if you don't want to spend all this time labeling camera trap images, maybe there are like some remotely sensed variables that could get at some of the variables on the ground that you're trying to think about. I don't know.
Yeah, so two ways of answering that. Firstly, we have got remotely sensed data for this area.
Yeah.
So at the moment, I've only looked at species richness, but like one of the things that I'll do after this is also look at forest cover and like land cover type, land use type, and see how that like corresponds to these images, because I think that will be really interesting to like see how, if we get any kind of metric, how that compares to other measures of like ecosystem health. And then the other, like in terms of multimodal data, is we've got so much acoustic data for this area as well. So I'm hoping to do a similar method for that. I mean, it will be much easier for acoustic data because it's already like in the season. Weirdly, acoustics is actually more advanced than camera traps in that area. So yeah, I'm actually quite excited to like see whether you see the same gradient in camera trap data than you do in acoustics, because it's like completely different animals. But yeah, I think the thing with like remote sensing is you're measuring very different things. So I think it's really interesting to use them to see how much they correlate. Yeah.
So one interesting thing too, we talked about this a bit, and I think it might be quite interesting, is I wrote a paper on active learning with camera trap data with Arash Nourizadeh, who's done also a lot of cool work with camera traps, and Dan Morris. And what we found is we were trying to figure out if there was a way to try to use the clustering and embedding space, and then just have a human label maybe one or two examples per cluster. and then use that to weakly label the cluster, and then use that to iteratively train and get good results for a new region without needing to label as much data. And we found that that was absolutely the case. You could like reduce the amount of training data you had to label by like 97%. But it was very important that the original embedding you used was trained with a triplet loss so that the distances in embedding space were maintained as opposed to being trained with a binary cross-entity loss. We got much better clustering from a camera trap model trained on mega detector crops of species, even in a different region with different species, if we trained it with a triplet loss and then applied it on a new region. So I think that probably a triplet loss trained model on these camera traps from like the Masai Mara, for example, might actually give you much better clusters.
Yeah, I think Omi did train, when he did this experiment with the Masai Mara data, he did use triplet loss and SimCLR. But I just used so I think I will talk to you more about that.
Because like a supervised triplet loss might be better informed than a SimCLR.
Right, yeah.
Because it actually has labels.
But
Did you try to see the dependence on the cameras? Like it's maybe the clustering is by like,
This is per camera.
oh, it's per camera?
what do you mean, like, did I color it by camera?
Oh, I was asking if you like if you looked at like, say, one camera trap and then all the species in there to see the distribution.
Oh, yeah, I did. I forgot to put that. Yeah, so I did like a quick look. Do you mean like, did I make out graphs for each one? And,
Like,
see
did you
how
just
the-
look at it?
Yeah, I did that for, like, I did just a quick example of like national park, a site from the national park, a site from the buffer zone, and a site from outside. And like, weirdly, the national park was really strongly clustered by day and night. So they had like two really strong, like really far apart clusters. Whereas like the buffer zone and outside buffer zone were way more just like no clusters at all, just like really like widely distributed. So that was quite interesting. So yeah, that's something that I'll look into a bit more as well. There's like a million different things that I need to look into and carry on. It's gonna be a never-ending topic.
Maybe one more question?
What do you think is a good temporal scale to be able to get a good biodiversity measure? Like, based on detectability, we'll be measuring biodiversity every like few weeks, months. What is a good temporal scale?
That's a good question. So this data set covers a month. I think it really depends on the environment that you're in, but like this data set covers a month. And we've got pretty much all the species that we would expect to see in our data set. So I think that's our limit. Like any less than I would it's a bit, I actually think sometimes a month isn't even enough, but to get like proper interesting questions about where the animals are. But yeah, I would say like minimum a month, but it really depends on your environment, because like some places you might not see, especially if you've got like hibernating species and things
It
that.
might also vary between months, but in the long term, you will see it change.
Yeah.
Thank you so much, Peggy.